T cells are immune cells that recognize and eliminate harmful cells by binding to proteins on their surface called antigens. CAR T cell therapy enhances this ability by genetically engineering T cells to express chimeric antigen receptors or CARs, which bind specific cancer antigens and activate cytotoxic killing. This approach has shown groundbreaking success against liquid blood cancers, reaching 80 to 90% remission in leukemia, but has limited success in solid cancers because they have a more diverse, heterogeneous population that expresses different antigens and can strip them over time. My hypothesis was that if tumor cells are genetically modified to overexpress the pan-cancer antigen CD276, then CAR T cell cytotoxicity in solid cancers can increase because it converts a heterogeneous tumor into a uniform population, which increases the likelihood of activating the CAR T cells. As you can see here, in a natural tumor, cells express varying levels of antigens, but if we genetically engineer them to all express a synthetic antigen, especially a pan-cancer antigen like CD276, which is expressed across many different types and especially for aggressive cancers, then they will all express high levels of the antigen and we can choose it as our CAR target. To test this, I designed a synthetic antigen combining CD276, an aggressive cancer antigen highly expressed across many cancers, with FLAG, a protein tag used to detect and track proteins. Then I designed a CAR receptor that is activated by both synthetic and natural CD276, also adding FLAG for detectability. To ensure proper CAR activation, I analyzed the change in protein shape and binding strength caused by the addition of FLAG. The data showed while there was minimal change in shape, there was still slightly reduced binding efficiency, but this may be beneficial because the affinity was still within the ideal range, and because natural CD276 binding is so strong, it could actually cause overactivation-induced exhaustion. Next, I isolated and amplified my synthetic CD276 gene from a bacterial plasmid, which is a little gene circle, so I could transfer the gene to a mammalian GFP plasmid, which allows cellular testing and helps visualize which cells have been genetically engineered through GFP, which makes cells glow. Then I found optimal DNA quantity needed to genetically modify the breast cancer cells with CD276 by lipoeffecting different concentrations of the mammalian GFP plasmid, selecting 2.5 micrograms as the optimal amount because it expressed more GFP fluorescence and had higher cell viability. Lastly, I programmed a simulation that compiled data to make a function that predicted CAR T cell cytotoxicity in breast cancer based on time, synthetic CD276 boost, and ratio of CAR T cells to cancer cells. The program proved that adding synthetic CD276 led to greater CAR T cell cytotoxicity. Collectively, these findings support synthetic antigen engineering as a promising strategy to overcome tumor antigen heterogeneity and antigen loss, two major barriers to effective CAR T cell therapy in solid tumors. In real-world applications, synthetic CD276 can be delivered to cancer cells through intratumoral injections of lipid nanoparticles carrying the antigen mRNA. With further validation, this approach may enable a universal CAR targeting strategy, reducing the need to engineer different CAR constructs for each tumor antigen type.